Hello, my name is Hannah Guss, and I am going to be talking about today about why late-stage parasitic disease control measures are failing and how we leverage whole genome sequencing to understand factors that contributed to schistosomiasis reemergence in China. Schistosomiasis is a neglected tropical disease which affects around 200 million people worldwide. It has been it's been in the process of being eradicated over several years, according to the WHO guidelines. However, China has come out as a key success in this elimination process and has successfully dropped down its number of eliminations to around less than 10,000. However, there is still pockets of reemergence that exist. Um, and despite very harsh control measures, there is still persistence in rural regions. To understand this better, we used whole genome sequencing of schistosomiasis myricidia, or the larval stage, to answer some main questions about how many distinct parasite lineages have contributed to reemergence, what does this imply about the number of independent introduction events, how have genomic patterns aligned with expectations for low prevalence, and have control measures and associated bottlenecks influenced parasite breeding, genomic variation, and selection. We have a total of 270 whole genome amplified Myricidia samples that were from 2007. These are archival samples that we whole genome amplified and then sequenced. From our results, we can see that the phylogenetic clustering demonstrates that village-based clustering, where samples from the same village tend to group together, and geographically close villages also show clustering patterns. However, some villages exhibit a more dispersed distribution, such as the villages S and R. However, this suggests a more complex genetic relationship than what we might have previously expected. We use NGS based relatedness analyses called RAB to uncover patterns of genetic connectivity. These are pairwise relatedness metrics that allow us to understand the amount of alleles shared between two different samples. We can see that some villages, such as village V, have very high mean relatedness, which suggests a very isolated lineage of schistosomiasis, while village F, for the other hand, has a very low relatedness, which may represent a more genetically admixed cluster. The heat map and the network shown here further reveal that the closely situated villages tend to cluster together, while the long-range villages links indicate possible historical or recent gene flow between distant populations. So, despite the populations being highly structured within the villages, there is some inter-village transmission, which shows that there is a failure of control measures to adequately stop transmission on a local scale. This phylogenetic analysis, these relatedness estimates show that parasites from the same village tend to be highly genetically similar, indicating that the village is primarily occurring at a micro scale within villages. This highlights the importance of household and village level interventions in breaking transmission cycles. Overall, our conclusions indicate that the highly localized patterns of genetic relatedness suggest human hosts within villages remain key drivers of transmission, allowing parasite populations to persist despite mass drug administration and other control efforts, such as cleaning up sanitation. Thank you so much for listening to my poster, and I'm happy to have any questions.